Unit 5 Human Physiology. The reductionist approach to study of life forms resulted in increasing use of physico-chemical concepts and techniques. Majority of these studies employed either surviving tissue model or straightway cell-free systems. An explosion of knowledge resulted in molecular biology. Molecular physiology became almost synonymous with biochemistry and biophysics. However, it is now being increasingly realized that neither a purely organismic approach nor a purely reductionist or living phenomena. Systems biology makes us believe that all living phenomena are emergent properties due to interaction among components of the system under the study. Regulatory network of molecules, supramolecular assemblies, cells, tissue, organisms, and indeed populations and the communities each create emergent properties. In this chapter, under this unit, major human physiological process like digestion, exchange of gases, blood circulation, locomotion, and movement are described in cellular and molecular terms. The last two chapters point to the coordination and regulation of the body events at the organismic level. Alfonso Corti, 1822 to 1888. Alfonso Corti, Italian anatomist, was born in 1822. Corti began his scientific career studying the cardiovascular system of reptiles. Later, he turned his attention to the mammalian auditory system. In 1851, he published a paper describing a structure located on the basilar membrane of the cochlea containing hair cells that convert sound vibrations into nerve impulse, the organ of Corti. He died in the year 1888. Chapter 16 Digestion and Absorption. Food is one of the basic requirements of all living organisms. The major components of our food are carbohydrates, proteins, and fats. Vitamins and minerals are also required in the small quantities. Food provides energy and organic materials for growth and repair of tissue. The water we take in plays an important role in the metabolic process and also prevent dehydration of the body. Bio macromolecules in the food cannot be utilized by our body in their original form. They have to be broken down and converted into simple substances in the digestive system. This process of conversion of complex food substances to simple absorbable forms is called digestion and is carried out by our digestive system by mechanical and biochemical methods. General organization of the human digestive systems is shown in the figure 16.1. 16.1 Digestive system. The human digestive system consists of the alimentary canal and the associated glands. Figure 16.1.1 Alimentary canal. The alimentary canal begins with an anterior opening, the mouth, and it opens out posteriorly through the anus. The mouth leads to the buccal cavity or oral cavity. The oral cavity has a number of teeth and the muscular tongue. Each tooth is embedded in the socket of jawbone. Figure 16.2. This type of attachment is called the thecodont. Majority of mammals, including human beings, forms two sets of teeth during their life. A set of temporary milk or deciduous teeth replaced by a set of permanent or adult teeth. This type of dentition is called the diffiodont. An adult human has 32 permanent teeth, which are of 4 different types in bracket heterodont dentition, namely incisors, canine, premolars, and molars. Arrangement of teeth in each half of the upper and the lower jaw in the order incisor, canine, premolars, molars is represented by a dental formula which in human is 2123 by 2123. The hard chewing surface of a teeth made up of enamel helps in the mastication of food. The tongue is a freely movable muscular organ attached to the floor of the oral cavity by frenulum. The upper surface of the tongue has small projections called papillae, some of which bear taste buds. Figure 16.1. The human digestive system. This is oral cavity, mouth, submaxillary and the sublingual glands. This is parotid glands, pharynx, esophagus, stomach, pancreas. This is liver, gallbladder, duodenum, transfer colon, ascending colon, ileum. And this is cecum and vermiform appendix. This is anus, rectum, sigmoid colon, descending colon and jejunum. The oral cavity leads into a short pharynx which serves as a common passage for food and air. The esophagus and the trachea in bracket windpipe opening into the pharynx. A cartilaginous flap called epiglottis prevent the entry of the food into glottis, opening of the windpipe during swallowing. The esophagus is a thin, long tube which extends posteriorly passing through the neck, thorax and diaphragm and leads to a J-shaped bag-like structure called stomach. The muscular sphincter gastroesophageal regulate the opening of esophagus into the stomach. The stomach located in the upper left portion of the abdominal cavity has four major parts: a cardiac portion into which the esophagus open, a fundic region, body, main central region and the pyloric portion which opens into the first part of a small intestine. Figure 16.3. The small intestine is distinguishable into three regions: a C-shaped duodenum, a long coiled middle portion jejunum and a highly coiled ileum. The opening of the stomach into the duodenum is guarded by the pyloric sphincter. Ileum opens into the large intestine. It consists of cecum, colon and rectum. Cecum is a small blind sac which hosts some symbiotic microorganism. A narrow finger-like tubular projection, the vermiform appendix, which is a vestigial organ, arises from the cecum. The cecum opens into the colon. The colon is divided into four parts: an ascending, a transverse, descending part and a sigmoid colon. The descending part opens into the rectum, which opens out through the anus. Figure 16.2. Arrangement of different types of teeth in the jaw on one side and the sockets on the other side. This is incisor, canine, premolars and molars, and this is socket of jaw. Figure 16.3. Anatomical region of human stomach. This is esophagus, fundus, cardiac, body, pyloric, and this is superior portion of the duodenum. The wall of alimentary canal from esophagus to rectum possesses four layers. In bracket figure 16.4, namely serosa, muscularis, submucosa and mucosa. Serosa is the outermost layer and is made up of the thin mesothelium in bracket epithelium of visceral organs with some connective tissue. Muscularis is formed by smooth muscles usually arranged into an inner circular and an outer longitudinal layer. An oblique muscles layer may be present in some region. The submucosal layer is formed of loose connective tissue containing nerve, blood and lymph vessels. In duodenum, glands are also present in submucosa. The innermost layer lining the lumen of alimentary canal is the mucosa. This layer forms irregular folds rugae in the stomach and the small finger-like foldings called villi in the small intestine. Figure 16.5. The cells lining the villi produce numerous microscopic projections called microvilli, giving a breast border appearance. These modifications increase the surface area enormously. Villi are supplied with a network of capillaries and large lymph vessels called lacteals. Mucosa epithelium has goblet cells which secrete mucus that help in the lubrication. Mucosa also forms glands in the stomach, gastric glands, and the crypts in the between the base of villi in the intestine, crypts of liver cone. All the four layers show the modification in different parts of the alimentary canal. Figure 16.4 Diagrammatic representation of transverse section of gut. This is serosa, this is inner circular and outer longitudinal. Both are called muscularies. This is submucosa, mucosa and lumen. Figure 16.5 A section of small intestinal mucosa showing villi. This is villi, this is lacteal and this is capillaries, arteries, crypts and veins. Figure 16.1.2 Digestive glands. The digestive glands associated with the alimentary canal include the salivary glands, the liver and the pancreas. The saliva is mainly produced by three pairs of salivary glands, the parotid in bracket cheek, the submaxillary, submandibular in bracket lower jaw and the sublingual in bracket below the tongue. These glands situated just outside the buccal cavity secrete salivary juice into the buccal cavity. Liver is the largest gland of the body, weighing about 1.2 to 1.5 kg in an adult human. It is situated in the abdominal cavity just below the diaphragm and has two lobes. The hepatic lobule are the structural and the functional units of liver, containing hepatic cells arranged in the form of cords. Each lobule is covered by a thin connective tissue sheath called the Gleason's capsules. The bile secreted by the hepatic cells passes through the hepatic duct and is stored and concentrated in the thin muscular sac called the gallbladder. The duct of the gallbladder in bracket cystic duct along with the hepatic duct from the liver forms the common bile duct in bracket figure 16.6. The bile duct and the pancreatic duct open together into the duodenum as the common hepatopancreatic duct, which is guarded by the sphincter called the sphincter of Oddi. The pancreas is a compound in bracket both exocrine and endocrine elongated organ situated between the limbs of C-shaped duodenum. The exocrine portion secretes an alkaline pancreatic juice containing enzymes and the endocrine portion secretes hormones, insulin and the glucagon. Figure 16.6 The duct system of liver, gallbladder and the pancreas. This is gallbladder and this is duct from the gallbladder. This is duct from the liver. This is common hepatic duct and this is common bile duct. This is pancreas, this is pancreatic duct and this is duodenum and this is hepatopancreatic duct. Figure 16.2 Digestion of food. The process of digestion is accomplished by mechanical and chemical process. The buccal cavity performs two major functions, mastication of food and facilitation of swallowing. The teeth and the tongue with the help of saliva masticates and the mix up the food thoroughly. Mucus in the saliva helps in the lubricating and adhering the masticated food particles into bolus. The bolus is then conveyed into the pharynx and then into the esophagus by swallowing or deglutition. The bolus further passes down through the esophagus by successive wave of the muscular contractions called peristalsis. The gastroesophageal sphincter controls the passage of the food into the stomach. The saliva secreted into the oral cavity contains electrolytes, sodium, potassium chloride, bicarbonate, and enzymes, salivary amylase and lysozyme. The chemical process of digestion is initiated in the oral cavity by hydrolytic actions of carbohydrate splitting enzyme and the salivary amylase. About 30% of starch is hydrolyzed here by this enzyme in bracket optimum pH 6.8 into disaccharide, that is maltose. Lysozyme present in the saliva act as an antibacterial agent that prevent infections. starch in presence of salivary amylase, and pH 6.8 gives out maltose. The mucosa of stomach has gastric glands. Gastric glands have three major types of cells, namely, first is mucous neck cells which secrete mucus. Second is peptic or chief cells which secrete the proenzyme pepsinogen. And third is parietal or oxantic cells which secrete HCl and intrinsic factor factor essential for absorption of vitamin B12. The stomach stores the food for 4 to 5 hours and the food mixes thoroughly with the acidic gastric juice of stomach by the churning movements of its muscular wall and is called the chyme. The proenzyme pepsinogen on exposure to hydrochloric acid gets converted into active enzyme pepsin, the proteolytic enzyme of the stomach. Pepsin converts proteins into proteases and peptones (peptides). The mucus and the bicarbonates present in the gastric juice play an important role in the lubrication and protection of mucosal epithelium from exoccretion by highly concentrated hydrochloric acid. HCl provides the acidic pH pH 1.8 optimal for pepsins. Rennin is a proteolytic enzyme found in the gastric juice of infants which help in the digestion of milk proteins. A small amount of the lipase are also secreted by gastric glands. Various types of the movement are generated by the muscularis layer of the small intestine. These movements help in the through mix up the food with the various secretions in the intestine and thereby facilitate digestion. The bile, pancreatic juice and the intestinal juice are the secretions released into the small intestine. Pancreatic juice and the bile are released through the hepatopancreatic duct. The pancreatic juice contains inactive enzymes trypsinogen, chymotrypsinogen, procarboxypeptidase, amylase, lipase, nucleases. Trypsinogen is activated by an enzyme enterokinase secreted by the intestinal mucosa into the active trypsin, which in turn activates the other enzyme in the pancreatic juice. The bile released into the duodenum contains bile pigments bilirubin and biliverdin, bile salts, cholesterols and the phospholipids, but no enzyme. Bile help in the emulsification of fats, that is breaking down of the fats into very small micelles. Bile also activate lipase. The intestinal mucosal epithelium has goblet cells which secrete mucus. Secretions of the breast border cells of the mucosa along with the secretions of the goblet cells constitute the intestinal juice or succus entericus. This juice contains a variety of enzymes like disaccharidase example maltase, dipeptidase, lipase, nucleosidase, etc. The mucus along with the bicarbonates from the pancreas protect the intestinal mucosa from acid as well as provide an alkaline medium pH 7.8 for enzymatic activities. Submucosal glands Brunner's gland also help in this. Proteins, proteose and the peptones partially hydrolyzed proteins in the chyme reaching the intestine are acted upon by the proteolytic enzymes of pancreatic juice as given below. Proteins, peptones, proteases in presence of trypsin, chymotrypsin and carboxypeptidase gives out dipeptides. Carbohydrates in the chyme are hydrolyzed by pancreatic amylase into disaccharides. Polysaccharides starch in presence of amylase gives out disaccharides. Fats are broken down by lipase with the help of bile into di and monoglycerides. Fats in presence of lipase gives out diglycerides and monoglycerides. Nucleases in the pancreatic juice acts on the nucleic acid to form the nucleotides and nucleosides. Nucleic acids in presence of nuclease gives out nucleotides and nucleosides. The enzyme in the succus entericus acts on the end product of the above reactions to form the respective simple absorbable forms. These final steps in the digestion occur very close to the mucosal epithelium cells of the intestine. Dipeptides in presence of dipeptidase gives out amino acids. Maltose in presence of maltase gives out glucose plus glucose. Lactase in presence of lactase gives out glucose plus galactose. Sucrose in presence of sucrase gives out glucose plus fructose. Nucleotides in presence of nucleotidase gives out nucleosides, and again in presence of nucleosidase gives sugar plus bases. Di and monoglycerides in presence of lipase gives fatty acid plus glycerols. The breakdown of bio macromolecules mentioned above occurs in the duodenum region of the small intestine. The simple substances thus formed are absorbed into the jejunum and ileum region of the small intestine. The undigested and unabsorbed substances are passed on to the large intestine. No significant digestive activity occurs in the large intestine. The functions of the large intestine are: first, absorption of some water, minerals and certain drugs. Second, secretion of the mucus which help in the adhering the waste (undigested particles) together and the lubricating it for an easy passage. The undigested and absorbed substances called feces enters into the cecum of the large intestine through ileocecal valve, which prevent the backflow of the fecal matter. It is temporarily stored in the rectum till defecation. Activities of the gastrointestinal tract are under neural and hormonal control for proper coordination of different parts. Sight, smell or the presence of food in the oral cavity can stimulate the secretion of saliva. Gastric and intestinal secretions are also similarly stimulated by neural signals. The muscular activities of the different parts of the alimentary canal can also be moderated by neural mechanisms, both local and through the CNS. Hormonal control of the secretions of digestive juice is carried out by local hormones produced by the gastric and the intestinal mucosa. Calorific value of protein, carbohydrates and fat (bauxite items not for evaluation). The energy requirements of animals and the energy content of the food are expressed in the terms of the measure of heat energy, because heat is the ultimate form of all energies. This is often measured to as calorie or joule, which is the amount of heat energy required to rise the temperature of 1 gram water by 1 degree Celsius. Since this value is tiny amount of energy, physiologists commonly use kilocalorie or kilojoule. 1 kilocalorie is the amount of energy required to rise the temperature of 1 kg of water by 1 degree Celsius. Nutritionists traditionally refer to kilocalorie as the calorie or joule (always capitalized), the amount of heat liberated from complete combustion of 1 gram food in a bomb calorimeter (a closed metal chamber filled with O2) is its gross calorific or gross energy value. The actual amount of energy combustion of 1 gram of food is the physiologic value of the food. Gross calorific value of the carbohydrates, proteins and fats are 4.1 kilocalorie per joule, 5.65 kilocalorie per joule and 9.45 kilocalorie per joule respectively, whereas their physiologic value are 4.0 kilocalorie per joule, 4.0 kilocalorie per joule and 9.0 kilocalorie per joule respectively. Figure 16.3 Absorption of digested products. Absorption is the process by which the end product of digestion pass through the intestinal mucosa into the blood or lymph. It is carried out by passive, active or phasolated transport mechanisms. A small amount of the monosaccharides like glucose, amino acids and some electrolytes like chlorides, ions are generally absorbed by simple diffusion. The passage of these substances into the blood depends upon the concentration gradients. However, some substances like glucose and amino acids are absorbed with the help of the carrier proteins. This mechanism is called the phasolated transport. Transport of water depends upon the osmotic gradient. Active transport occurs against the concentration gradient and hence requires energy. Various nutrients like amino acid, monosaccharides like glucose, electrolytes like sodium plus are absorbed into the blood by this mechanism. Fatty acids and glycerol being insoluble and cannot be absorbed into the blood. They are first incorporated into small droplets called the micelles, which move into the intestinal mucosa. They are reformed into very small proteins coated fat globules called the chylomicrons, which are transported into the lymph vessels (lacteals) in the villi. These lymph vessels ultimately release the absorbed substances into the blood stream. Absorption of substances takes place into different parts of the alimentary canal like mouth, stomach, small intestine and large intestine. However, maximum absorption occurs in the small intestine. A summary of absorption (site of absorption and substances absorbed) is given in Table 16.1. Table 16.1. The summary of absorption in different parts of the digestive system. Mouth. Certain drugs coming in contact with the mucosa of the mouth and the lower side of the tongue are absorbed into the blood capillaries lining them. Stomach. Absorption of water, simple sugars and alcohol, etc. takes place. Small intestine. Principal organ for absorption of nutrition. The digestion is completed here and the final product of digestion such as glucose, fructose, fatty acids, glycerol and amino acids are absorbed through the mucosa into the blood stream and the lymph. Large intestine. Absorption of water, some minerals and drugs takes place. The absorbed substances finally reach the tissue which utilize them for their activities. This process is called assimilations. The digestive waste solidified into coherent feces into the rectum initiate a neural reflex causing an urge or desire for its removal. The ejection of feces to the outside through the anal opening defecations is a voluntary process and is carried out by the mass peristalsis movements. Figure 16.4. Disorders of digestive system. The inflammation of the intestinal tract is the most common element due to bacterial or viral infections. The infections are also caused by the parasites of the intestine like tapeworm, roundworm, threadworm, hookworm, pinworm, etc. Jaundice. The liver is affected. Skin and eyes turn yellow due to deposit of bile pigments. Vomiting. It is ejection of stomach contents through the mouth. This reflex action is controlled by the vomit center in the medulla. A feeling of nausea precedes vomiting. Diarrhea. The abnormal frequency of bowel movements and the increased liquidity of the fecal discharge is known as diarrhea. It reduces the absorption of food. Constipation. In constipation, the feces are retained within the colon as the bowel movements occur irregularly. Indigestion. In this condition, the food is not properly digested, leading to the feeling of fullness. The causes of indigestion are inadequate enzyme secretion, anxiety, food poisoning, overeating and spicy food. PEM, that is protein energy malnutrition. Dietary deficiencies of proteins and the total food calories are widespread in many underdeveloped countries of the South and Southeast Asia, South America and West and Central Africa. Protein energy malnutrition, that is PEM, may affect large sections of the population during drought, famine and political turmoil. This happened in the Bangladesh during the Liberation War and in Ethiopia during the severe drought in mid-18th. PEM affect infants and children to produce marasmus and kwashiorkor. Marasmus is produced by a simultaneous deficiency of proteins and calories. It is found in infants less than a year in age. If mother's milk is replaced too early by other foods, which are poor in both proteins and calorific value. This often happens if the mother has second pregnancy or childbirth when the older infant is still too young. In marasmus, protein deficiency impairs growth and replacement of tissue proteins. Extreme emaciation of the body and thinning of the limbs result. The skin becomes dry, thin, wrinkled. Growth rate and the body weight decline considerable. Even growth and the development of brain and the mental faculties are impaired. Kwashiorkor is produced by protein deficiency unaccompanied by calorie deficiency. It results from replacement of mother's milk by a high calorie, low protein diet in a child more than 1 year in age. Like marasmus, kwashiorkor shows wasting of muscles, thinning of limbs, failure of the growth and the brain development. But unlike marasmus, some fat is still left under the skin. Moreover, extensive edema and the swelling of the body parts are seen. Summary: the digestive system of humans consists of an alimentary canal and the associated digestive glands. The alimentary canal consists of the mouth, buccal cavity, pharynx, esophagus, stomach, small intestine, large intestine, rectum and the anus. The accessory digestive glands include the salivary glands, the liver (with gallbladder) and the pancreas. Inside the mouth, the teeth masticate the food, the tongue tastes the food and manipulates it for proper mastication by mixing with the saliva. Saliva contains a starch digestive enzymes, salivary amylase, that digests the starch and converts it into maltose, that is disaccharide. The food then passes into the pharynx and enters the esophagus into the form of bolus, which is further carried down through the esophagus by peristalsis into the stomach. In stomach, mainly protein digestion takes place. Absorption of simple sugars, alcohols and medicines also takes place in the stomach. The chyme (food) enters into the duodenum portion of the small intestine and is acted on by the pancreatic juice, bile and finally by the enzymes in the succus entericus, so that the digestion of carbohydrates, proteins and fats is completed. The food then enters into the jejunum and the ileum portion of the small intestine. Carbohydrates are digested and converted into monosaccharides like glucose. Proteins are finally broken down into amino acids. The fats are converted to fatty acids and glycerols. The digested end products are absorbed into the body through the epithelial lining of the intestinal villi. The undigested food feces enters into the cecum of the large intestine through ileocecal valve, which prevents the backflow of the fecal matter. Most of the water is absorbed into the large intestine. The undigested food becomes semi-solid in the nature and then enters into the rectum, anal canal, and is finally egested out through the anus.